DNA unit. DNA is deoxyribonucleic acid. It stores genetic information and it produces, well, it dictates protein production. It is double-stranded. Nucleic acid are nucleotide. It has nucleic acid which are nucleotide subunits. Until 1950, it was believed that protein was a basic genetic material of a cell, and Alfred Hershey and Martha Chase did some research on that. Deoxyribonucleic acid stores genetic information, and the Hershey and Chase experiment proved this. Biologists hesitate to accept DNA as molecules of heredity. Knew more about proteins than we did about nucleic acids, and proteins had 20 building blocks, or amino acids, while DNA had just 4 building blocks, nucleotides. In 1950, Alfred Hensley and Martha Chase conclusively proved that DNA and protein, and not protein, is a genetic material. The chemical structure is double-stranded, and in the early 1950s, Rosalind Franklin and Maurice Wilkins proved this. X-rays of DNA revealed regularly repeating structure of the building blocks. In 1953, James Watson and Francis Crick determined double helix structure of DNA. Complementary bases are held together by hydrogen bonds. Twisted ladder. Rails are deoxyribose sugar plus phosphate backbone. They have 5-inch, 5-foot end, and 3-foot end. It's some kind of unit. Well, it uses foot as a unit, but I don't really know exactly what it means. I just know it's 5-end and 3-end. Length, whatever. The rungs are nitrogenous bases. Adenine always pairs with thymine, thymine, adenine, guanine, cytosine, cytosine, guanine. Nucleic acid have nucleotide subunits. The chemical structure: purines, adenines, and guanines. Pyrimidines are cytosine and thymine, and hydrogen bonds hold them together. An organism's genome is all genetic material in its cells. Prokaryotic genome consists of one circular DNA molecule. Eukaryote genome is divided among multiple chromosomes housed in the nucleus. Each chromosome is a package of DNA and associated proteins. Chromosomes. Each chromosome is a discrete package of DNA coiled around histones and other proteins. Protein synthesis, translation, and transcription. Transcription takes place in the nucleus. Cell rewrites genes' DNA sequence to a complementary RNA molecule. Translation takes place in the endoplasmic reticulum. Information in RNA is used to assemble a different class of molecule, protein. RNA, ribonucleic acid. Nucleotides contain sugar ribose instead of deoxyribose. Has nitrogenous base. Uracil binds with adenine. Has base ACGU. No thymine, single-stranded. Can catalyze chemical reactions. 3 types of RNA interact to synthesize protein: mRNA, rRNA, and tRNA, or messenger RNA, receiver RNA, and transfer RNA. Transcription, DNA template used to build RNA. And damn, this only took 15 minutes to type. And 3 minutes to read out.